This presentation shows you how to prepare a direct wet mount of a fecal specimen. Fecal samples can be examined for the presence of protozoa as well as eggs and larvae of helminths. In order to identify these parasite stages using the microscope, the sample of feces has to be prepared properly. There are several different ways to prepare fecal specimens for microscopic examination, and the direct fecal wet mount procedure you will learn here is one of the easiest and basic techniques used in a parasitology lab. We can prepare wet mounts using a fresh fecal sample. This is called a direct wet mount. These basic direct wet mounts are prepared using saline or iodine. We can also prepare wet mounts from concentrated fecal specimens. So there are two types of wet mounts: the direct wet mount prepared using saline or iodine. The other wet mount is prepared from a sample of concentrated feces. You need to examine at least 3 direct wet mounts before the results of a fecal specimen is reported negative for parasites. As I mentioned before, microscopic observation of a direct wet mount is one of the simplest and most widely used method to detect parasites in feces. This technique is useful to identify parasites to the species level. In protozoa, the trophozoites and cysts may be demonstrated and identified. In helminths, eggs and or larvae may be observed and identified using a direct wet smear. Direct wet mounts are very helpful to make a quick diagnosis of heavily infected specimens. Laboratory safety is an important aspect when handling fecal specimens. You must treat all fecal specimens as potentially infectious and do observe the recommended actions shown on the screen when working with fecal samples. After watching this presentation and doing the exercises, we expect you to be able to do the following actions shown on the screen. In other words, these are the learning outcomes of this exercise. The main materials needed for this procedure are shown here. Let us look at how to prepare a saline wet mount. A saline wet mount is used as the initial step in the microscopic examination of feces. It is useful to demonstrate trophozoites and cysts of protozoa and also eggs and larvae of helminths. Saline mounts will reveal motility of protozoan trophozoites and also show if ingested red blood cells are present. First, take a clean microscope slide. Place a drop of saline in the center of the slide. The saline should be at room temperature for observation of trophozoite movement. Using a toothpick, take a small amount of stool and place it on the drop of saline. The fecal sample should be fresh if you are to see motile parasite stages. Make sure you sample from inside as well as from the surface of the fecal sample. When sampling, do not take too much of the fecal specimen. The size of a matchstick head is more than sufficient for this procedure. Mix the stool in the drop of saline to make an even suspension. Carefully place a cover slip on the drop of saline. In order to avoid air bubbles in the smear, hold the cover slip at an angle and touch the edge of the saline drop and then carefully lower it onto the slide. If you see excess liquid, press the cover slip gently and remove the liquid that comes out by dabbing with a piece of tissue. A good saline smear should not be too thick or too thin. A good smear will be uniform in appearance. It should not be too thick that fecal debris can obscure parasite stages. It should also not be too thin that there are blank spaces. A good fecal smear should be thin enough to just allow newsprint to be read through, as shown on the screen. If you have taken too much sample, your smear will be too thick and it will be very difficult to observe parasite stages among the fecal matter. Now you can examine the preparation using the microscope. Place the slide on the microscope stage and focus on the saline mound. Adjust the light in the microscope field as appropriate. Because too much or too little light will make your examination and detection of parasite stages difficult. First, focus using the 10 times objective and scan the whole smear systematically to find any parasite stages present. In order to scan systematically, you can start at the top left-hand corner of the cover slip. Then move the stage systematically from left to right or up and down as shown in this diagram to scan the whole cover slip area. You must also focus up and down to see all layers of the specimen. In addition, take effort to examine each microscope field carefully before moving to the next field. Your examination must be systematic to reduce the chances of overlooking any stages present. If suspicious objects are seen, then examine with the 40 times objective to see the more detailed morphology. The use of an oil immersion objective on wet mounts is not routinely recommended. The helminth stages are easily detected and identified using a saline mount. Usually, the helminth eggs and larvae are large enough to be seen using the low power magnification. So use the 10 times objective. A few helminths may need 40 times objective. Helminth eggs and larvae will not need any staining. Now you should also know the characteristic features to identify the stages to the species level. For instance, the size and shape are important species characteristics. Also, the stage of development is important. That is, whether the egg has one cell or many, or whether it is embryonated and containing a larva are things to observe. In some, the thickness or color of the shell can also be used as a diagnostic feature. Then some eggs contain special structures like an operculum, spines, mammillated layer, hooklets, etc., which will help you to identify the species of the helminth. Larval stages of helminths, like the larvae of Strongyloides stercoralis, may also be detected in a saline mount. If the stool sample is stale, you might also see hookworm larvae. In fresh stool samples, you will not see hookworm larvae. So, if the stool sample is stale, it is important that you should be able to differentiate between the Strongyloides and hookworm larvae by examining their mouth openings and the genital primordium. In saline wet mounts, you will be able to see trophozoites and cysts of protozoa like amoebae, flagellates and ciliates. Saline mounts are also useful to detect motile protozoan trophozoites. As you know, motility is one of the diagnostic features of protozoan parasites. To detect and identify most protozoans, you will usually need a high-power objective. With the 40 times objective, you will be able to see the motility, shape, structural elements, and many of the inclusions like ingested red blood cells, chromatoid bodies, but detailed structure of the nucleus will not be clear with 40 times objective. Usually with an iodine preparation, we can confirm the diagnosis of protozoan cysts. However, sometimes it may be necessary to make permanent stain smears and measure the size to ensure the correct identification. The shape, size, motility of trophozoites, nucleus, inclusions in the cytoplasm, special structural elements, etc. are important in the species identification of protozoa. Lugol's iodine is used for the temporary staining of parasite stages, especially the protozoa. Iodine will kill any organisms present, so no motility will be seen in trophozoites after iodine is added. To stain with iodine, take the saline mount prepared. Take 1 to 2 drops of Lugol's iodine solution and touch the pipette to the edge of the cover slip. You will see the stain gradually diffusing into the saline preparation. You can also prepare a new slide by mixing the stool with the drop of iodine. In an iodine mount, the cytoplasm will stain yellow or light brown, and the nuclei and glycogen masses will stain dark brown. You must examine the iodine preparation within 5 to 15 minutes of making the smear. If left for too long, the parasites will overstain and make the identification difficult. As in any technique, there are limitations in the direct wet mount procedure as well. As we mentioned previously, direct saline mounts are very useful to detect parasites in heavily infected samples. But if the number of organisms in the sample is low, direct wet mounts may not detect them. That is, there are chances to overlook the parasitic organisms. It may also be difficult to identify protozoa which are very small to the species level with just the direct saline wet smear technique. Motility is very helpful to identify protozoa and trophozoites, but this characteristic motility is lost when placed in iodine. So direct wet mounts are very helpful to make a quick diagnosis of heavily infected specimens. But because of these limitations, results obtained from wet smears have to be confirmed by concentration and permanent stained smears.